Right? So when we look at our secondary structure, that's really what we're concerned about, is how can the structure orient itself such that we can minimize the polarity of those two bonds. We want to minimize that oxygen's negative, and we want to minimize that positive on that hydrogen by aligning them so that they can cancel out with other side chains. Right? So we're looking at hydrogen bonding between or in our backbone. Right? The next big thing that gets mentioned, which does play a really important role, but we're not going to talk about it too much, and that's what this figure is trying to show. If we go through and look at that backbone, we do have the ability to rotate about our carbon with the R group. Right, we can't rotate about the peptide bond, because what's the issue with the peptide bond? It is resonance stabilized. Just to kind of address this. So if we took a look down the peptide bond, where that's our carbon, and then we got our nitrogen in front, with our pi bond out to our oxygen here, and then our carbon out to our R group. If we take a look at that nitrogen, it is technically sp3 hybridized. We got the lone pair hanging off of one of those bonds. This is the favorite orientation. Why is it locked in this type of Newman projection? All right, and as some people addressed, there is some steric hindrance here. These carbons get darn close to each other. Right, likewise, our hydrogen and oxygen get darn close to each other. Why would we favor that orientation as opposed to, say, rotating it and putting our largest group, our carbon, down at the bottom?
Because you have even distribution on angles?
Nope. 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 What are you saying about electrons? Maybe?
If you rotate it right, they're going to interfere with the oxygen.
Not quite. Close.
The electrons are being equally shared by the oxygen and the carbon?
Yes. Remember, the amide bond is stabilized through resonance. For there to be resonance, the pi electrons must be parallel to the lone pair. If they are not in parallel planes, the electrons cannot be shared. When we take a look at this Newman projection, where's our p orbital for our oxygen and our carbon? It's in that plane. If we take a look at our sp3 hybrid orbital for our nitrogen, parallel orbitals. As soon as we rotate about that carbon-nitrogen bond, this orbital now swings up to horizontal. Is it now parallel to the pi system? Nope. If it's not parallel to the pi system, it cannot contribute to resonance. Okay, so we lose stability of our amide bond if we rotate out of that. Everybody see that? Okay, so it's kind of an interesting way of looking at it. You guys had actually already mentioned the stability of the amide bond via resonance, so I figured I'd see if you guys could project that out back into the orbital diagrams. And that's where that question came from on the test. Yes?
So there's a hydrogen bond right here between a hydrogen and oxygen, right?
That distance gets darn close. It's not quite close enough to allow for hydrogen
Okay,
bonding.
that's what I was curious, because I thought there was, and I was like, why is it rotating?
So you can get some interactions there.
Yeah.
But we want to try and maximize that. And so what we'll do is orient a little bit closer, or rotate our structure, rotate about that bond, our carbon, nitrogen and our carbon-carbonyl bond, okay, because we can rotate about those. That rotation, because those are the only two bonds that we can rotate around, for some reason they decided to name these. Those are, shoot, I don't know that Greek letter. That one's psi, that one's, is that theta?
Theta.
Okay. Psi and theta are how those angles or those bonds get referred to. So if we rotate about those, we end up generating new angles. Okay. Depending on what secondary structure we get very particular angles. Are you responsible for knowing what those exact angles are? Absolutely not. Okay. You are responsible for understanding that those angles can rotate, and that's where the structure is going to adjust to allow us to get into the secondary structure. Instead of referring to those as psi and theta, we can also refer to them, let's see, it's on here, oh, we already mentioned hydrogen bonding. Our Ramachandran angles, okay, those are your psi and theta. Okay, your psi is going about the carbon-carbon bond, and your theta is going about the carbon-nitrogen bond. There are a variety of plots that you could go through and look at to see where those angles are and how they orient. Okay, again, depending on what type of secondary structure you get, you'll get very particular angles from those bonds. Okay, so just kind of an interesting effect. The force, we already addressed this. What was our force? Way to all volunteer at once. We're looking at hydrogen bonding. Okay, so it's the hydrogen bonding generated from our backbone that's going to allow us to take on certain shapes. So what are those shapes? Okay. If our backbone interactions occur over a short distance, what we're going to look at is something known as an alpha helix, which is shown on the left. And apparently we've got some stacking issues with the text. Or if they happen over kind of a longer distance, we look at beta sheets. Okay? It is still a hydrogen bond, which means the interaction is still narrowed down to a very particular distance. So what are we referring to when we say short versus long? It's not the length of the hydrogen bond, but it's the length or the distance between what atoms are actually causing our hydrogen bonding interaction. In the alpha helix, our hydrogen bonding action is happening very quickly within a couple sequences or a couple amino acids within the straight chain. Okay, so when we look at the alpha helix, we're looking at a hydrogen bonding interaction happening, got it on the next couple slides, I think 4 amino acids away. Okay? So what ends up happening is we rotate about those psi and theta bonds and generate this helical shape. That helical shape brings down another amino acid darn close to a previous one, and we get that hydrogen bonding interaction. What we're talking about when we talk about long distance interactions is now it's not just that one immediately next to it or 4 amino acids away. We could be looking at 20 amino acids away. We could be actually looking at an entirely different polypeptide chain. But what we're concerned about is how those chains orient with respect to each other. Okay? So in our beta sheets, what ends up happening is it's a sequence of amino acids that have aligned perfectly and another sequence that is then stacked exactly with it. Okay, so it's 2 sequences of amino acids running parallel or technically anti-parallel to each other. And that's how we generate those 2 main motifs. Okay, so common folding patterns that we would see within proteins. Okay? Questions about those 2?
So basically the alpha is just like one single strand.
Yep.
The other ones have multiple ones.
Have the potential to be multiple ones. So if we take a look at a long strand, okay, it can coil on itself. There's an alpha helix. But if I take a look at that same long strand, I can still get beta sheets. Instead of it coiling, now let's just fold it over.
Okay.
And fold it over.
Okay.
Okay. And it could very well be that I have no beta sheets through the middle, but I have a beta sheet at the very ends that then fold over on top of each other. Okay. So the beta sheets are, if you look at modeling, tend to be a little bit less stable. The coiling tends to be a lot more structurally sound and stays in that general shape. Now I think about it, I think I left out a cool video that shows a lot of this. If we take a closer look at our alpha helix, okay, the images that you're going to see in your textbook are not the easiest to pick out, and that's because what we're trying to do is draw a 3-dimensional structure. Okay, if you think back to chairs, how easy was it to manipulate your chairs? Okay, your cyclohexane chairs in organic chemistry. Some people picked that up pretty well, some people didn't. What we're looking at here is now a much more complicated version of that. We effectively have chairs on top of chairs, and then you have to predict how each of those chairs interacts with each of the other chairs. Okay, so it becomes a lot more complicated to view and ultimately draw. So instead of looking at it as a stationary object, let's see if this is going to work for me. Okay. What we can go through and do is look at it in 3 dimensions. So what they're showing here is that yellow structure being your, well, it's going a lot faster than I can talk about it. The yellow was your alpha helix. They kind of zoomed in and simplified it. And now they're showing the side chains in green, and then your amino groups aligning with your carbonyls. So because of how that structure is shaped, it can force that interaction so those 2 atoms get very close to each other. Okay, and hopefully in a second we can zoom back out add in the rest of the structure, notice that all of your side chains aim outwards, okay, away from the alpha helix. If we turn and look from the other direction, we can now see the core, or the center, right through the middle of our alpha helix. We've got the hydrogen bonding all the way down it, and as you rotate, and if you look really carefully, you may have to do it at home, you can see dotted lines connecting each of those. So as they flip and rotate this molecule, you can start to see that shape. That shape is exceedingly difficult to see, so in future 3D modeling, what we end up doing is converting it into just a coil. So we can see the coil a little bit cleaner than we can other things. All right, so if you think back to that ATP structure that we worked with, that structure, sorry, if we shift back, a little bit inelegant, that structure had a whole bunch of these weird coil-looking shapes. Those weird coil-looking shapes were alpha helices. Okay, so what we're looking at is those individual interactions. So what conditions might prevent our alpha helix from forming?
pH.
Okay, if we adjust the pH, is that going to affect our backbone? It's only going to affect the side chains. Remember, within your backbone, you don't have any acid-base characteristics. Okay, so it might mess with the hydrogen bonding ability, okay, but it's not really going to do too much beyond that. So it might tweak it, but our alpha helix system is so stacked that we're probably not going to see much with the pH.
Temperature?
If we change the temperature, how is that going to affect it?
It rotates more, so
We get more molecular vibrations. The more vibrations we can generate, the more we can actually cause those interactions to break down and try and find other favorable interactions. So we can start to break things with heat. Okay, oh, that's temperature as well. Okay.
Order of amino acids.
We could look at the sequence of amino acids, in particular what amino acids are in that alpha helix. Why would that matter?
Because it's the same reason you get beta sheets. If it's in a certain sequence, then you'll get. So usually if you insert something into there that's not in the helix, then it could change.
That can't fit within the helix. Is there an amino acid that has an odder or a more different shape than some of the others? Proline. If we put proline in there, that actually causes a kink in the amino acids sequence. That's going to potentially prevent this side chain interaction or the backbone interaction because it's going to cause that primary structure to kind of bend and tweak in an odd fashion and prevent that psi versus theta bond angle change. So we can look at the primary structure affecting this as well. Okay, and I said at least three, but I definitely didn't write them down. The next slide doesn't show it, so let's just take a look at what other things we can look at. There's a whole bunch of random facts that you can remember with your helices. We'll see clockwise or right-handed helix or helices. Why might we not expect left-handed? Okay, so when we generate a helix, what we're looking at is ultimately a screw. Okay, an inclined plane wrapped around a helix or a cylinder. Right, which means we've got directionality with that screw, with the threading on that. So what we're looking at here is clockwise. Why would we expect to see only clockwise and not counterclockwise?
Because the proton pump only
Right,
moves in
sorry.
one direction.
No.
I'm just kidding. And then it's
Because they're
Well,
made in
that's
the-
true, but that's going to be something else. What's that?
Is it because they're made in the northern hemisphere?
No.
Because the left,
That's the baby joke.
left-handed can then take a lot more heat. It's usually found
D
in-
and L. We've got chirality with each of your amino acids.
Yeah.
That chirality is going to force a particular chirality with the helices. Okay. So if we change the chirality, instead of having, we typically have L amino acids, if we made them all D amino acids, we might predict that we'd get the opposite helix. Okay, because it's how those individual side chains change the overall structure. Okay. Within an individual turn, so as we kind of rotate up, to get that full rotation, we're looking at 3.6 amino acids. It's not perfect because it ends up winding a little bit tighter than 4 amino acids. Okay, yes, that is an important thing for these. The repeat distance, so if we look from one, ultimately sine wave, if you wanted to look at it that way, because if we look at it, whoops, that was the wrong button. If we look at a helice, right, that distance, when we come back to matching, come on now, did I just lose connection? How could I have lost connection?
Once we finish writing this down.
Oh, what I was doing was trying to draw so we could see that. There it goes. So if we looked at it in 2 dimensions, it just looks like a sine wave. The distance in that repeat is your 5.6 angstroms. Okay. Whoops. And that's angstroms, not a capital A. What's the difference between capital A and angstrom? Yeah, there's a little dot on top of that. I forgot to go back and fix that symbol. So we're looking at 5.6 angstroms for that to occur. Each amino acid is hydrogen bonded to the 4th amino acid that is distant from it. That kind of has to go back with how many amino acids per turn, our 3.6. So if we count out our amino acids and try to see where that hydrogen bonding interaction is occurring, if we start at 1, that one's going to hydrogen bond to the 4th amino acid away. That's all that's trying to say. And then our hydrogen bond is going to be parallel to the coil axis. Where's the coil axis in this drawing? Right up the middle. We're wrapping effectively around a pencil, if you will. Okay, so your hydrogen bond interaction is in that same axis. And then, as that figure nicely showed, or that movie, all our R groups face outwards. Why can't they face inwards? It's too tight. Our coil is wound way too tight. If we're going to do that, it's just not going to fit. I wouldn't be able to fit those amino acids in there.
Are even the hydrophobic R groups sticking out?
All R groups, period, in an alpha helix face outwards. So that could be another question. If all of those R groups happen to be hydrophobic, where would we expect to find the alpha helix? On the
Same spot.
interior of our protein. If they're hydrophobic, we don't want to be out on the exterior where there's water. Okay, so that's if it's a water-soluble protein. Is there some place in a cell that is not hydrophilic? In the membrane. So if we take a look at an alpha helix and we notice that all the R groups are hydrophobic, we could predict that that protein would be found, or that portion of the protein would be found very buried inside itself, okay, away from water, or it could be found in a membrane. Because in the membrane, we're looking at a very hydrophobic interaction anyway. Maya? Questions about your alpha helices? Only it's just to list a bunch of facts. Our beta sheets, okay, this is where we're taking a look at individual strands that can then align with each other. So what we're trying to show in each of these cases is a new strand, or potentially new strand, that's aligning with another strand. Okay. You'll notice that we can end up with something known as parallel or anti-parallel. What are they referring to? Okay, well, yeah, direction, but what's that directionality associated with? Yeah, with our hands it actually works. If we said my fingertips was a nitrogen, okay, and then my wrist was going to be, or my elbow was going to be the carboxylic acid end of the, okay, we can look at parallel with another sequence lining up exactly the same, or anti-parallel. Okay. So it's looking at how those beta sheets end up forming. Okay. What happens to our R groups? Where do you think the R groups are going to be aimed? What do you mean by outside?
Outward.
I still don't know what outward means.
Like if the sheet's like this, it's going to be this way or this way.
Yeah, so if we call the sheet here flat, and I know what you were trying to get at. If we say our beta sheet is in the plane, okay, of our paper, or the projection here, our R groups are going to be facing either out towards you or inwards. And in fact, it's going to alternate. Okay? One will be outwards, the next one will be inwards.
Is that only for the anti-parallel, or can that be also parallel?
It's both. Okay. They will end up maintaining this conformation, this S trans and planar. What is it? So the planar hopefully makes sense. Our amino acids are all planar, okay, right through that backbone. What's the S trans referring to?
Because you said like one goes out.
That the rotation and then the.
Kind
one,
of.
One on the opposite side.
So if we look at an amino acid,
Did that stair shape? Like how it goes this way?
yeah, if we refer to where we have any double bond characteristic, which is where? It's at our amide bond. Okay, what is the alignment of the two groups around that amide bond? Opposite sides, also known as trans. What if it was S cis?
It'll be close, yeah.
Okay, and then our chain builds out that way. Oops, no, that's our group, sorry. Okay. Those of you might remember this. If you think back, at least those of you from Mesa, if you think back to your Diels-Alder reaction, take a look at that background information on your Diels-Alder reactions. Which conformation allows for a Diels-Alder reaction to occur the fastest? The S cis. There's a conversion between your S cis and S trans. So that nomenclature is ultimately coming from organic as well. In this case, it's again in reference to where we have the most double bond characteristic.
The S refers to like a chiral.
The S refers to what type of bond is technically between. Oops. There are all sorts of problems with that. Let's fix that to appropriate configuration.
Sigma.
And that's why I have that extra carbon. Okay. That is a sigma bond officially. Sigma begins with S. There's the S of your S trans versus your S cis. It's referring to the sigma bond that has the most double bond characteristic, which is why we can bring in that trans. Okay.
So it will always be in that combination?
It's always going to be in the S trans conformation, and you can see why that's going to be necessary if we go through and look at the S cis conformation.
And it can flip between parallel and anti-parallel, right?
Yep. So the parallel anti-parallel just has to do with how those chains align with each other. Okay. The S cis is what's happening in the chain. Okay. For our chain to occur, for it to get into this overlapping situation, we have to have it have this jagged line. Okay. Those are all trans. If I go through and switch one of those, see if I can do this appropriately, to cis, what happens? Oops. We'd get a bend in it, which means I can't do a beta sheet wrapping. Okay, so that's the issue with that. We're always going to have that S trans conformation to allow for the beta sheet formation. As soon as we make it S cis, we put a kink in it and it can't align a beta sheet anymore.
Can be R trans?
R trans?
S trans can be R trans?
S doesn't refer to R and S. S refers to sigma bond. We're looking at the sigma bond in our peptide. Other questions on that? Your carbonyl and your nitrogen will be perpendicular to the axis of the sheet, okay, which if we go through and take a look at this, we're looking at our sheets running up and down in our drawings. I'm going to have to erase everything. If we look at our nitrogen and our carbonyl, okay, they're parallel, or sorry, that's not parallel. That's perpendicular to the axis of our sheet, which is running up and down. Okay. So the hydrogen bonding interaction occurs perpendicular to the axis of the beta sheet. You guys see that? Okay. Hydrogen bonding has to occur between the carbonyl and the nitrogen. What color is a carbonyl?
Okay, I see it now. Red.
Yeah, your carbonyl, you're looking at that red atom. Your nitrogen is the blue. Your hydrogen bonding interaction occurs between those all the way through. The beta sheet runs up and down. Okay, everybody see it now? So ultimately all that's happening is we're breaking down each of these structures. Remember our primary structure was just the sequence of the amino acids, which we said was important because if we change out an amino acid, we can change the potential interactions within that protein. And that's one of the things that happens with our sickle cell anemia. We change out one amino acid for something that has drastically different chemistry. Okay? That drastic difference prevents the particular interactions that we would need to allow for that protein to function properly, even though the protein itself may still fold appropriately. Right? Secondary structure is looking at now how we take that amino acid, or sorry, not our amino acids, our peptide chain, that sequence of amino acids, and how do those amino acids start to interact with each other? Okay? How does the backbone of that chain potentially interact? And that's what's generating these different shapes. Okay? And that's where we get our alpha helices and our beta helices, or sorry, beta sheets. Okay, so that's all that our secondary structure is really referring to. Okay. As far as our parallel versus anti-parallel, this shows it a lot better. If we take a look at that top sequence, we're running nitrogen to carbon, right to left. If the sheet it's interacting with is running that same general sequence, our nitrogen to carboxylic acid end, okay, we're looking at parallel sheets. What happens in the bottom situation? If we change the orientation of our amino acids or our peptide sequence, we're now running anti-parallel. Yeah? That's a good question. I don't think so. Okay, it's going to have to do with the interactions that you end up finding. You end up with the same number of interactions. It looks to me kind of like the anti-parallel looks neater.
That's what I was
So you could see something along those lines that your anti-parallel allows for more compact packing. More compact packing generally means more stable. Okay.
Wouldn't the parallel be more compact since the two chains are actually closer together?
They might be able to push closer, but you might see the distance being odd in that. So I don't know exactly how it's going to work as far as those exact distances. Okay. We already addressed our S-trans. The R group, where's our R group? Okay, coming out and back.
Wouldn't there be hydrogens to attach to those carbons?
So all they've shown here is so far the amino acid sequence without specifying any R groups or hydrogens. The carbon dead center, take a look at that carbon, that looks roughly dead center, has a hydrogen and an R group. The carbon next to it, does it have anything attached to it? No hydrogen. Why no hydrogen?
Carbonyl.
It's a carbonyl carbon-oxygen
The one
bond.
next to it?
Which direction next to it, left or right?
Left.
If we go to the left, it's again going to have a hydrogen and an R group.
That would have the little steric hindrance, wouldn't it?
That has steric hindrance if the R group aims into the beta sheet. Where does our R group aim? It aims out. So it comes either out or back. We don't have to worry about that steric
And they
hindrance.
alternate, right?
And they're going to have to alternate.
That's why the anti-parallel is a bit closer.
No, remember, your R group's
Oh,
coming
okay.
out and back. It has nothing to do through the middle.
Okay.
Okay. Other questions? So if we're going to take a look at our beta sheets, okay, we could potentially see these things occur in the same structure, particularly the anti-parallel. So if we take a look at our nitrogen, we could be going with our sequence all the way through the middle here. And then right at this end, it could U-turn and come back the other direction. Okay. To be able to do that U-turn, there's a couple things that might be necessary. Number one, what's one option? Okay. We could put an amino acid that forces a sharp turn on it. What amino acid forces a sharp turn? Proline. So we have a high tendency of seeing proline out there. Okay. What other things could show up out there?
S-cis on the,
What's that?
you have S-cis on the end.
So we could looking at changing the bond angle in there. That's true, and that will have to happen. Okay, so we could be looking at an S-cis change in that orientation, which makes sense.
Maybe a basic amino acid,
Why basic?
because it's gonna accept one of the other hydrogens or pull on it at least.
In that turn, you're now flaying that side chain way far away from the rest of it. So you're probably not going to see any side chain interactions there.
So I mean like small, like when, then like have like sulfur, disulfur bond.
So you said two things there. You said small, agree.
Like,
Then you said valine.
no, like yeah, small like valine.
Valine's got 3 carbons. That's pretty big.
Or like
There we go.
okay,
We can switch to glycine with a single hydrogen. Glycine sterically has very small things in the way. So glycine is also a likely amino acid to see in that U-turn. Good call.
then like disulfur bridge.
And then you talking about disulfur bridges, where would we see disulfur bridges?
Alpha.
In cysteine amino acids. Would we expect to see that though? Remember where your R groups. One's up, one's down. Can I get a cysteine bridge? No, it's going to be distance-wise too far away. Okay, so you got the right idea on potentially causing it to fold back on itself, but in the beta sheet, that's going to be a bit difficult to set that up. All right, so when we're looking at those U-turns, do I not have the structure? Oh, I just animated it in. So what we're looking at is likely seeing the amino acid of proline, okay, or glycine. Proline because it actually forces the kink. Why glycine? It's just small, can allow for that. There's another thing that we could expect to see out in that U-turn.
Too, right?
Alanine's pretty small too. We could potentially see that. So we'd be looking for smaller amino acids. Does it have to be just a few amino acids? No, it could actually be a huge long chain of amino acids that then doubles back to allow for the beta sheet to form. Okay? So it doesn't have to immediately double back on itself. If it does immediately double back, we're looking at either small amino acids with relatively small side chains so that they don't sterically block and prevent the folding back, or we can look at proline because that will help force the amino acids or force that chain to double back on itself a little bit more quickly. Okay? You see how knowing your amino acids could actually start to help predicting structures? This is why I want you to go through or spent some time having you memorize some of these. Okay. When we're looking at secondary structure, is it only alpha helices and beta sheets? There's a lot more, technically. Come on. We could look at a whole bunch of other types of secondary structure. Secondary structure is ultimately looking at how these things kind of interact and fold with each other. Depending on what you're looking at, you might see other shapes. So in this case, what is happening with my spacing? In our image on the far left here, okay, what does that abbreviation look like we're forming? What interactions do we have here?
It looks like a beta sheet.
We have anti-parallel beta sheets, but a beta sheet we'd expect to be a sheet. It's not exactly a sheet. It's actually coiled up on itself. So it's almost a beta sheet helix. Okay? So that's a new type of motif on how these things are coming together. That's typically known as a, I want to call it barrel roll, but I think that's not right. I think it's I'm pretty sure it's a beta roll. Okay? So what we're looking at is it folding back on itself. Okay? There are other options. Okay? We can look at tighter versions of it, or what's happening out here. We have alpha helices and beta sheets interacting with each other. Okay? And we typically don't classify these things as our secondary structure, because what we're starting to look at is how our alpha and beta secondary structures start to interact with each other. So we're starting to look at another order of magnitude of these interactions, which means we're ultimately looking at tertiary. Okay? So we're in kind of a weird gray area, and the textbook addresses this as well. You're not looking at something that's strictly secondary, but you're not looking at something that we would typically classify as tertiary. So that's why I've labeled it as kind of a secondary plus. Okay? You're kind of fitting in that gray in-between space between those interactions. There isn't a clear cutoff line on these. Okay? So as long as someone goes through and sees common repeating patterns, they can refer to it as a structural motif. That then typically gets classified as secondary structure. Okay. So what is tertiary structure? Tertiary structure is taking a look at how your secondary structure interacts, but then as the entire protein. Okay, the entire sequence of the amino acid. Within each of these cases, I could potentially have my amino acid come out here, go through another alpha helice, do a couple beta sheets, all that kind of fun stuff. Now that entire picture is my tertiary structure. But if I focus just on that role, now I'm looking at the secondary structure. Okay, so it's kind of that gray area between, is it the entire protein that we're looking at to describe what we're seeing, or is it just a section of the protein, regardless of how many amino acids are involved in that? Kind of makes sense? Okay. Where might we see these kind of roles? Okay, shown from the top, what do you see right through the middle? A hole. Where might we have holes, or want proteins that have holes? In membranes. If we want something to be able to pass through our membrane, okay, to get up to that surface, it should be hydrophilic, because that's our water surface, but then to be able to pass through, the middle of the membrane is all hydrophobic. So if it comes up hydrophilic, and it may attach the surface of the membrane, but how can it possibly pass through? You're going to have a hydrophilic, hydrophobic repulsion, and it won't be able to make it through the membrane. Okay? So we need to have some way to allow things to pass through, okay? Particularly in a controlled fashion. These types of protein folds can allow for porosity, can allow things to pass through.
So porosity means?
Yeah, porous. And
I've just never heard of it.
I may have made up porosity as a word, but I'm referring to porous.
It's like, wow,
It sounds really legit.
Yeah.
It does. The sheets always roll.
Sheets do not always roll, okay? And that's why this is typically classified as a secondary structure. Okay? Your beta sheets typically just align flat, depending on like a sheet. Like a
Primary?
sheet is secondary.
Secondary.
The sequence of amino acids is primary structure.
Is it kind of like graphite?
Yep,
So if you ask, like, if beta rolled secondary or?
I would expect secondary. Yeah. Okay. If you're really on kind of a gray area, yeah, it's still one thing. It's not referring to the entire protein. If I said the entire protein folds into a beta roll, then what is it?
I guess tertiary.
Now we're going to say tertiary because it's the entire protein. And so that's the gray area between it. Secondary is just referring to a chunk of amino acids that fold into a very particular structure, okay, a motif. The tertiary structure is how does the entire sequence of amino acids within your protein fold.
I thought it could be all the same thing.
Sorry?
Could it mean if it's all the same combination, it only has beta sheets, then it's still,
Yeah, if the entire structure is all beta sheets,
but you still have to know the entire structure.
yeah, you'd have to specify that entire structure to get your tertiary classification on that.
Okay.
If you're just referring to a sequence of amino acids within the entire primary structure, then you're looking at secondary. How do those amino acids interact with each other? Is that another question?
So for the alpha and beta sheet, does it have to be in parallel for that structure?
For this one to happen? I don't know that. That just happens to be this motif. I wouldn't be surprised if somebody found it, your beta sheet's going in anti-parallel, and they now decided that it's a new structural motif.
Okay.
Why would they decide that?
Those are anti-parallel?
Because their name gets attached to it.
Okay.
Isn't that not anti-parallel?
This one's anti-parallel, but does it have the alpha helices going
Oh,
with
okay,
it?
I get you.
Okay. Other questions with our kind of secondary plus structures? So when we push, and this is kind of what we're all already talking about, going in between secondary versus tertiary. Secondary is common structural or repeating motifs. Okay? So how does that individual sequence of amino acids fold to generate a new shape? Not saying the entire protein. Our tertiary is now saying we take the entire protein and we look at how it folds as a whole. Okay? So that is going to include the secondary structure, because how those individual secondary structures interact with each other is going to now set up your tertiary, how the whole thing gets put together. Okay? So it's a very kind of gray area between those. We end up with two primary categories, but when we're looking at our tertiary structure, we end up with fibrous proteins and globular proteins. Would have been nice if I'd stepped. No, I didn't give you this information. So our fibrous proteins, you can look at collagen. Fibroid?
Fibrogen.
I was going to say, I didn't think I spelled that right. I think that's supposed to be fibrogen.
Fibroid.
Yeah, I don't know what happened there. Keratin, all of those are fibrous proteins. If we look at globular proteins, think ATP synthase, okay, the one we've already been evaluating, okay, how it all kind of chunks together. So if we look at our fibrous proteins, we're looking at things that sequence in kind of a strand, okay, long, strandy, stringy things. Our globular proteins, you're looking at balls, okay, so we're just looking at how just the proteins all, come on now, it wasn't funny that time. Okay, those proteins just kind of wrap up on each other. What would we expect for solubility between these two? Okay, would we expect a difference first off? Yeah.
Fibrous is empty though.
Where would we, so if we take a look at our fibrous proteins, what are our solubilities for those? Very, very little. Okay, what are we using our fibrous proteins for? Structure. Okay, do we want our structure to be water soluble? No, because if it dissolves in water, it now falls apart and we don't have the structure anymore. So our fibrous proteins have very minimal solubility. What kind of amino acids would we expect in those? Hydrophobic amino acids, so that they don't interact with water well. Right? If we shift to our globular proteins, okay, globular kind of run the gamut between being water soluble and not, but in general, we typically think about them being water soluble, at least more so than our fibrous proteins. Okay? And that's because those things are now floating through a solution that have to move from one place to another. And that whole transportation, when we look at a cell, is ultimately going through a very polar water-type environment. So the solubility of your globular proteins tends to be much more water soluble. What amino acids might we expect to find on our globular proteins? Kind of a trick question. It's going to be a little bit of both. The inside of the globular protein we would expect to be hydrophobic, and the outside should be hydrophilic, okay, depending on where it's located.
I was just thinking about like ATP synthase.
And again, depending on where ATP synthase, the parts of ATP synthase were located, we're going to end up with different solubilities.
I was just thinking like one's inside the cell.
One's inside the membrane, one's outside the membrane. Yep. All right. So what's going to allow all this protein fold? Man, these images are horrible. What happened with the lecture? What did I do? Oh, that's really weird. So what your textbook has done is actually giving you a rather nice image, and that's that image that probably the only thing we can see. What we're looking at in that case is all the different types of interactions that come into tertiary structure. So you'll notice that we do end up with this kind of helice running through it, okay, which is our alpha helix. You'll notice that we also have a sequence doubling back on itself. So we do have that beta sheet as well. But further, okay, as a bigger addition with our tertiary structure, we can see the introduction of other things. Okay, we have a metal coordinating to particular amino acids. For metal coordination, what amino acids would we expect to find interacting with a metal? Ionic ones, but you can take it further than that. The acidic amino acids will typically interact with metals because what charge are the acidic? They're negatively charged on a neutral pH, and what charge is our metal ion? Positive. So we would expect to find our acidic amino acids very, very close to metal ions.
So is the metal ion kind of like mimicking the beta sheet?
So it can allow and force folding,
Okay.
yes. And that's one of the things that makes our tertiary structure more interesting, is that if we incorporate these other things, that can cause things to bend on themselves. Within a beta sheet, okay, on that fold back, we might expect to find even a metal in that pocket.
So that could be something that causes.
That helps that bending process, yep. If we look way down in the side, inside here, I know they've got it labeled, but we can pretend we don't see that. They list leucine, valine, and isoleucine. Right, what amino acids? What's the physical property of those? They are all hydrophobic interactions. So we would expect our hydrophobic amino acids to all fold up very, very close to each other because they want to minimize their interaction with our polar water solvent. So we end up seeing a hydrophobic interaction showing up in our tertiary structure. Okay? And as Victor was also mentioning, we can also see something known as, why did I not draw? A disulfide bridge, right? Which is going to happen between cysteine molecules or cysteine amino acids, because the side chain of our cysteine gives us that CH3 and our sulfur. So we can form that sulfur-sulfur bond. Right? If we're looking at a long chain and we've now linked them back to each other, again, we can force that bend by doing that linkage. We're now branching, we're connecting those two things together. If you think to polymer chemistry, which you probably haven't heard anything about, it's a form of cross-linking. We've cross-linked two branches and we brought them back to each other through a new bond. That's that disulfide bond. Yes?
So can we include the metal ion as another thing that forces the bend? U-turn?
Yes. The metal is something that can force a U-turn. The disulfide potentially as well. That just gets a little bit tricky in the general structure. Okay? In a tight bend, it's probably not going to work. Same issue with the metal. In a tight bend, it's probably not going to work. Okay? But in something that had a few extra amino acids there, we could probably get that to double back.
Is this how they made like something to alleviate heavy metal poisoning?
So heavy metal poisoning is an entirely separate issue. With heavy metal poisoning, what you need is something to bind to the heavy metal that isn't your body and then be able to dispose of it. Okay? In that case, you're looking at something known as a ligand. If you think back to at least MESA, EDTA, oxalic acid, all of those are ligands because they can bind to metal. The biggest issue with most ligands is they are not selective for a particular metal. So older people tend to get kind of roped into this snake oil treatment of EDTA chelation. Oh, you got to get rid of all those toxic metals by taking this chelator. So the chelator just grabs metals. Fun thing about EDTA, it does not select for toxic metals. It selects for all metals. Are there metals that are important to standard protein binding? Yeah. If we pull all those metals out, yeah, you may not have any metals in your body. Good job. You now don't have any proteins that are folded appropriately.